Okay guys, welcome back to Unit 9 Video B. We got a couple of white blood cells left to talk about before we talk about a little bit of physiology. So let's keep going. The lymphocyte, this is going to be the second most abundant of the white blood cells. And this cell will develop in the bone marrow, but it'll continue to develop and it'll also mature in the lymphoid tissue. Now we're going to talk about the lymphatic system in another unit, so I'll save that talk for then. They can further develop, or we can still break them down in a couple of categories called T lymphocytes and B lymphocytes, and that'll be pretty clear when we talk about the immune response, what the difference between the T cells are and the B cells. Okay, these were the lymphocytes. These will be the white blood cells that you'll see most common in a viral infection. So cold, this type of cell would make up most of the whole blood, or sorry, the complete blood count. We can have atypical forms, but don't worry too much about that. But how we characterize them is by having, they're relatively small for a white blood cell, and the nucleus takes up a large part of the volume of the cell. Okay, the last white blood cell we're going to talk about now is the monocyte. The monocyte are usually the biggest white blood cells that you'll see in a blood smear. So they're characterized as being the largest. They'll have an irregular shaped nuclei, and you'll be occasionally under high magnification, you'll be able to see vacuoles in amongst the cytoplasm. These can form into macrophages that can help envelop invaders. So these can form into cells that will go around through the blood, and they'll act as cells that engulf anything that's foreign. So bacteria or viruses are there, they'll go around and they'll be the ones that'll phagocytose them, bring them inside the cell where they can destroy them by using lysosomes and enzymes and stuff like that. If you're ever having issues with your immune system, for example, if you were to take cancer drugs, chemotherapy, those drugs are usually designed to essentially eliminate your immune system white blood cells. These are the first type of cells that will come back after cancer treatment. Okay, that's it for the white blood cells. The last of the formed elements we have to talk about now are the platelets. These are the smallest of the formed elements. They're not true cells. They don't have a nuclei like the red blood cells. There's no DNA or anything like that present. They're fragments of cells. Don't worry about the megakaryocytes. That's not something we're going to get into. But they're basically cell fragments. They're much smaller than even the red blood cells. But they do occur in higher numbers than the white blood cells. So they're kind of in the middle in terms of numbers. We had 4.5 to 5 million red blood cells per microliter. We had around, a couple of thousand, up to 10,000 white blood cells per microliter. And now we've got about 150 to 450,000 cells in a thrombocytes in a microliter. So that's where we stand, about an order of magnitude below the red blood cells, and maybe a couple of order of magnitude above the white blood cells. Now, the function of the platelets is to move around the blood, the circulatory system, but if there's any damage in any of the blood vessels, these guys are the ones that are going to stick to it. Okay, so they'll stick to any damage or breaks in the lining of the blood vessels, and they'll form these platelet plugs that will prevent any loss of blood outside of the circulatory system. Alright, so let's talk a little bit now about some physiology. The first process we're going to talk about in this unit is hemostasis. Now, if we were to break that word down, hemo, of course, is going to talk about blood. And stasis, we've already talked about stasis, we've seen that before, but it means to stay still. So hemostasis is the process that prevents blood loss, okay? So it keeps our blood within our circulatory system, so it doesn't escape. So if a blood vessel is ruptured, if you have any bleeding, hemostasis is what's going to prevent that and hopefully return our blood back to normal levels. There's three steps to this process. First, we have contraction of the smooth muscles in the blood vessels. So we said we've got these smooth muscles that line all of our blood vessels in our body, and when these are damaged, or a blood vessel is damaged, these muscles will constrict, and we call that vasoconstriction. Vaso is going to refer to the blood vessels, and constriction just means to get tighter or get smaller. So that's our first step, number one, vasoconstriction. Once that happens, when we're going to have our platelets that are circulating, they're going to stick to the damaged part of the blood vessel, and they're going to form that platelet plug, and that's the second step. And then third, we get the formation of a blood clot, and that's what's going to kind of seal that blood vessel so that blood doesn't escape anymore, and the cells there present near the injury can start to rejuvenate or renew, and then start the healing process. Now, coagulation is basically when a bunch of proteins and cells stick together and they form a bigger unit. So it requires a bunch of reactions that lead to the formation of a protein called fibrin. So fibrin is kind of an insoluble protein that's kind of sticky. It kind of forms a net, and that protein net will trap blood cells, and it'll cause the formation of a clot. Coagulation results from a lot of different factors that are present in the plasma of the blood. There's going to be lots of different clotting factors there. Ions, for example, calcium plays an important role in this. When tissue are damaged, often these clotting factors are released, and that helps promote quicker blood control, so we get less loss of blood. And there's even going to be enzyme precursors. So there's going to be molecules that are released that will eventually turn into enzymes that will cause reactions to help the clotting process or the coagulation process. So the final steps of coagulation, this is going to be the first real physiology that we're talking about, is going to be in these stages right here. Okay, we're going to have a molecule called prothrombinase. So look at this word, pro, and then ase at the end. Pro we've seen before when we talked about prokaryotes. So if you see pro at the front of a word, it usually means before. Okay, so prokaryote means before nucleus. If you see a word with the last three letters are going to be ase, this is going to be an enzyme. So this molecule here, prothrombinase, is an enzyme, but it's not quite ready to function yet. So prothrombinase is circulating around in the blood, in the plasma, and when there's damage to the plasma and we need to have coagulation occur, prothrombinase will, in conjunction with calcium, will turn into prothrombin. Prothrombin is still a precursor. So prothrombin will then turn into thrombin. Thrombin will then react with another protein called fibrinogen, and that produces yet another protein called fibrin. Now fibrin, if we look down here in the picture down here, is going to be these green filaments down here. This green is going to be fibrin filaments, and as you can see, it kind of forms a network or a net, and it catches platelets, it catches red blood cells, and that's going to help form that sticky plug that's going to prevent any blood loss through the blood vessel. So if we go back up to the top here, we've got our blood vessel right here. First we'll have the vasoconstriction. The blood vessel will get smaller. Then we'll get the platelets will find the damaged part, and they'll start to stick to it and form this platelet plug. And then we'll start going through this process of physiology, where we start off with prothrombinase, and prothrombinase will turn into prothombin. Then it uses calcium to turn into thrombin, and when thrombin's present, it can help turn fibrinogen into fibrin, and then those fibrin threads will trap more platelets, more red blood cells, and it'll essentially stop any bleeding. So it's a little bit of a complicated process, but that's what happens when we start to have coagulation, and we end up with stopped a bleed. Alright, so let's talk about blood type. Now, we talked about blood type in the lab before, and we went through it pretty good. I think from everyone's marks, everyone did quite well. So you guys know now that blood type is based on antigens that are on the surface of the red blood cells. And those antigens have, they respond to antibodies that we can find in the plasma of cells. And of course, we talked about what happens when we mix incompatible blood types. So here's our table. I believe I actually showed you this table in the lab. So it tells us how to tell the difference between blood types. So here we've got blood type A, B, AB, and O. And those are the 4 blood types we can get from the AB blood group. So if we just take blood group A, for example, then that's going to have the A antigens on the surface of the red blood cells. It'll have anti-B antigens in the plasma. Okay? If we look at blood type B, those are the ones that'll have B antigens on the surface of the red blood cells, and they'll have A antibodies in the plasma. Blood type AB is going to have both A and B antigens on the surface of the red blood cells, but they won't have any antibodies in their plasma. And then blood type O, the final one here, won't have any antigens. There's no A or B antigens on the surface of the red blood cells, but they have both anti-A and anti-B antibodies in the blood plasma. So you can see here in the table, it tells you what groups these groups, different blood groups, can donate to. It's a good idea to understand how this works, but I don't think you need to memorize this table, for example. Okay, so when we test for blood type, and we can't do that in the lab anymore just for safety precautions, what we do is we take a sample of your blood, of course, and we mix it with anti-A or anti-B antibodies. If we get a coagulation with the anti-A, then we know you're blood type A. If we get coagulation with anti-B, then we know you're blood type B. If there's no coagulation at all, then we know that you are blood type O. The second thing we always test for is the Rh factor, which is going to be the A positive or A negative. And the Rh factor is tested in the exact same way. If there's coagulation, it's positive. If there's no coagulation, it's a negative test. So here's a look at the same type of things we talked about in the lab. If you're anti-A and you clot with the anti-A, then we know you have type A blood. So this circle right here is showing coagulation of red blood cells. This is here showing a normal red blood cells. So here's what what type A blood looks like. Here is what we'd see with type B blood. This is type AB, and then finally no coagulation at all is going to be type O. So this is just showing you some of the blood compatibility tests. We talked about this before. We have a universal donor. Patients with blood type O can donate to anybody because there's no antigens on the surface of their red blood cells. So even if you're A, B, or AB and you've got antibodies in your plasma, it can't attack those cells because there's no antigens to attack. A patients can donate to blood type A and AB. B patients can donate to B and AB. And AB patients, of course, can only donate to AB. These guys, however, are the universal recipient because they don't have any antibodies in their plasma. So it doesn't matter what blood cell we give them, there's no antibodies there to attack the red blood cells. So blood type O negative is the universal donor, and blood type AB is the universal recipient. Now, the Rh factor is another thing we can work into the blood group, and that gives us the blood type, the positive or the negative. We can come across cases in pregnant women where we can have an incompatibility between the mother and the newborn. Now, this can lead to a condition called hemolytic disease of the newborn. Now, look at the word hemolytic. Hemo will mean blood. Lytic means to split. So in this condition here, red blood cells will be broken and they'll come apart, and that can be serious to the baby. So what happens is we get a mother with her first pregnancy who's Rh negative and the baby is Rh positive. During birth, they'll have some mixing in the blood. Now the mother's blood will recognize the Rh factor as an antigen, so it will attack it. When it does that, it'll produce the antibodies for anti-B.